Hello friends, in this video we will discuss about transport of Auxin. Okay friends, there are two types of transportation in the plant Auxin. Auxin transportation, there are two types of. First one is polar transport, and second one is non-polar transport. In polar transport, the Auxin transport by the cell to cell manner, and in non-polar transport, Auxin travel with the help of phloem. Now we will discuss briefly about the non-polar transport and polar transport of Auxin. In polar transport having active transport, that means the active transport which is energy dependent transportation, whereas non-polar transportation is passive transport, which is energy independent. That means that there is no requirement of energy. Now polar transport is slow and short distance. It travels on very short distance, whereas non-polar transport is very rapid and long distance transport, transportation. Okay, now we will discuss briefly about the polar transportation, which is cell to cell manner. There are so many types, special types of proteins required for the transportation. We will discuss in the diagrammatically. Okay, let us consider we make cell to cell manner here. This one is plasma membrane, and this is the cell wall of plant, and now this one is another cell where transportation occurs. We know that there are so many special pores here, which is connectivity which is known as junction, which is known as plasmodesmatter. This one is, we all of us know that this connectivity is connection of cell to cell is known as plasmodesmatter. Plasmodesmatter. Now here we draw another cell also. Auxin is mostly two form. Chemically, auxin is known as I discussed in previous lecture also. Chemically also known as IAA. That means the indole acetic acid. Acetic acid. There are two another types of naturally occurring indole auxins are IBA and PAA. IAA is mostly present in all the species of plant, okay, and IBA and PAA is also a natural auxin. IAA means indole acetic acid, IBA means indole butyric acid, and PAA means phenyl acetic acid. Okay, here we take indole acetic acid inside the indole acetic acid which is present in this cell in the bound form, that is known as IAAH. That is H means the protonated form of IAA. This one is bound form. It breaks down to form ions IAA and H plus ion. Okay, and some proteins present here transporter protein present here which is known as pin protein. Okay, pin protein, with the help of this, proteins come at the apoplastic, IAA plus comes at the apoplastic region. This is apoplastic region. We all of us know that cells of plant bound by the plasma membrane, firstly plasma membrane, after then this one is cell wall, and in both of them having between both of them having a space which is known as apoplast region. Okay, and in this region, apoplastic region, IAA comes. Okay, this transporter protein is known as pin protein. is also known, this protein is also known as anion carrier protein. Anion carrier protein. Okay. Pin protein is also known as anion carrier protein. There are so many different types of pin proteins are present. Later we will discuss. First, IAA minus this anion form of auxin comes at the apoplastic region, and from the plasmodesmata it will come here. It comes here. IAA minus. Okay, and now the IAA is combined with the hydrogen ion. Okay, both are combined to form bound form of IAA H, like this. Okay, IAAH. Now this IAA work as they comes directly inside the cytoplasm of next cell. This IAA is lipophilic in nature, whereas this one is IAA minus is not a lipophilic in nature. That is why IAA minus do not cross this plasma membrane. Okay. Now IAA minus, which is also come, IAA minus is also come without bounded form with the help of transporter protein, which is known as pin protein. Sorry, this protein is not. This protein is pin protein, and this protein is AUX-LAX1 protein. This is the AUX-LAX1 protein. With the help of this protein, IAA minus comes inside the cytoplasmic region. Here IAA H, which is bounded by the proton, and also comes in cytoplasm, and IAA minus, which is not a lipophilic protein but comes inside the cytoplasm with the help of AUX-LAX1 protein. Here the pH is raised. pH is about 7, and whereas here the pH is about 5. That is why most of the IAA bound with the hydrogen ion, just because of hydrogen ion concentration at the apoplastic region is high. Now, but some IAA comes with the IAA which is free form. comes with the help of AUX-LAX1 protein. When auxin comes here, then rest of the auxin which is bounded by the proton, which also separate. Hydrogen ion is come here and IAA is here. Now the IAA minus, that means the auxin is free form. Here also IAA minus at free form, and here also IAA minus is in free form. This reaction is also we can see inside this protein. Okay, inside this cell. Here both the auxin comes like this, and here is also a plasma membrane, plasmodesmata. Now this IAA and this IAA comes here to transport in next cell. And here this reaction also takes place, and then transportation cell to cell manner occurs. This one is now this hydrogen ion comes inside the apoplastic region, comes at the apoplastic region with the help of hydrogen pump. Here ATP is used to transport this hydrogen ion to the apoplastic region. This type of hydrogen ion present all the regions of plasma membrane. Okay, then the hydrogen ion concentration increases in this region, apoplastic region. Then the pH is reduces like this. Okay, now this is all about the transportation of auxin, transportation of auxin, which is polar transportation. Okay, this is all about transportation. Some proteins we will discuss here. Some proteins, pin proteins, about the pin proteins like pin 1 and pin 4 protein help in the basipetal transportation. But that means the transportation to the shoot region to the root region, and basipetal transportation. Basipetal transport. That means the auxin transport to the base region, and pin 3 and pin 7 also help to transport laterally. Lateral movement of transport occurs with the help of pin 3 and pin 7. And pin 2 also help in the transportation of auxin to the base, to the upper region. That means the acropetal transportation. Only this protein work at only elongation zone of root. But now what proteins are further moved towards upper region of protein, that is ABC B type, B type 4 B type 4 protein also help in the transportation from the base to the upper region of plant. And ABCB, that means the ATP. What is the meaning of ABC? ABC means ATP binding cascade, superfamily B. Okay, ABC binding cassettes, superfamily B. This is the family of protein belonging to the ABC family, which is ABC B type family. Okay, having 4. And laterally ABC lateral movement, another protein also help, ABC B type 19 help in lateral movement. There are so many proteins, ABC B1, ABC B4 is also here, ABC 14, ABC B type 14, ABC B type 15 is also discovered here. Okay. Now we will here hydrogen ion present, which help in the expansion of what? Expansion of cell. Because one of the protein here present, which is known as expansion protein, which help expansion of cell. That is why auxin is responsible for elongation of shoot tip. Okay, here expansion protein help in expansion in the presence of hydrogen ion. That means that this protein active at the acidic region. Okay, acidic region, or when auxin comes here, then this expansion protein help in the expansion. Okay, in the presence of acidic environment at the apoplastic region. This expansion protein actually do not break the covalent bond of myofibrils. Do not break the covalent bond of myofibril. What is myofibril? Myofibril present in cell wall region. Okay. It breaks the cell wall. That is why expansion takes place. But there is it do not break the covalent bonds of cell walls. Okay. Another fungal toxin which is known as fucococin. This one is fungal toxin. It is also help in the activation of hydrogen pump. It is also help in the activation of hydrogen pump. That is why fucococin help in the expansion of shoot tip. Okay, fucococin actually activate hydrogen ion pump. When hydrogen ion pump active, then hydrogen concentration here increases. When hydrogen concentration increases, here makes acidic environment where expansion protein present. Expansion protein breaks the cell wall. Okay. In the two form. What is the two form? That is directly. It can directly IAA, that means the auxin, directly transport and indirectly transport. Directly means when auxin attach here some cell surface receptor. Okay, cell surface receptor. Let us draw here. Let us draw here. We draw again this. I want to explain you signaling, how IAA signals to makes this region acidic. Okay, this a little bit we will draw here. This is the plasma membrane. And this one is cell wall of the cell. Here, now this is, let us consider this one is cell surface receptor. Cell surface receptor where auxin attached. Auxin attached here. Okay. Here secondary messenger occurs, and which secondary messenger receive this signaling. This signaling, and this signaling activate hydrogen ion pump, which is responsible for the this one activated and which is responsible for the hydrogen ion comes here. This one is of IAA minus. Okay. When or and another, if IAA is bounded form, then here it will come because of it is a lipophytic protein. Lipophytic form. Okay, that is why it do not require any type of receptor, cell surface receptor. That is why it crosses the plasma membrane of cell and it come here. It comes here. It comes here like this, and it bound with ISM. ISM means intracellular signal molecule. It binds with the intracellular signal molecule. Now, we all of us know that here present nucleus where DNA is also occur, and this signaling activate regulatory protein, and this regulatory protein comes inside the nucleus where it activate transcription factor of hydrogen ion pump, where it consider it is hydrogen ion pump gene, and it transcribe to form mRNA, and then it will mRNA translate to form protein, which is hydrogen ion protein, hydrogen ion pump protein, and this transportation comes here. This protein comes here. Consider this makes like this. This one is hydrogen ion protein pump, which is transport to the plasma membrane, and it also activate this. When once hydrogen ion pump of plasma membrane activate, then the hydrogen ion comes here. Therefore, this expansion protein work here for the expansion. This is little bit signaling of IA, how they are work in polar transport. Okay, it is all about the polar transportation. Okay, and non-polar transportation not a very difficult thing. I have already discussed that non-polar transport is passive transport and energy independent. Energy independent process, and it is very rapid process, long distance process, and it transport by the phloem, whereas phloem transportation occurs in the cell to cell manner, and it is an active transport, energy dependent and slow and short distance process. This one is efflux. efflux protein, and this is the this one is pin protein, which is known as influx protein. Okay, then this is all about the transport of auxin. Thank you, friends. When you like this video, when you understood about the transportation of auxin, then please subscribe my channel Lucky's Biology Tutorial. It will absolutely help. This lecture is absolutely help in the CSIR as well as ARS net. Okay, thank you so much.